recorded. So we are recording now, 7:43, which means we have about 17 minutes before we get started. So if you're watching the video, probably skip ahead about 15 minutes or 17, whatever. It's Covington Grid, right?
No, sir, Marshawn.
Oh, yeah, sorry. Gotcha. A little extra credit for being so early. Top of whatever extra credit, whatever else extra credit I get today. I'm just up here checking emails, so if you have any questions, concerns, you know. Excuse me. Morning. Morning. All right, less than 2 minutes left. Start attendance. I like going first. Bangura, not here yet. Case Bolt, not here yet. Tristan, not here yet. Covington Griggs, not here yet. Gately, not here yet. Dow, not here yet. Hughes, not here yet. Lebdy, not here yet. Carla, not here yet. Mankins is here. Mathis, not here yet. Rains is here. Thank you. Reed is online. Don't forget, if you're online, you have to send in the words before 9 a.m. to get the credit and to be considered present. Elise is here. Thompson, not here yet. Woodrow, oops, not here. Dewey, not here yet. Elkins is here, correct? Yes, Elkins is here. And Dow just came in, correct? Is there anybody I missed? All right, so keep in mind, guys, extra attendance is just extra credit. I mean, it's mandatory according to the university. And I do submit attendance. As a matter of fact, the next time I have to submit it will be this coming up Monday. So don't get too upset. I know there's one person that had, at least one person that had some issues with attendance, or not issues, singular issue. I made a mistake because sometimes when I'm calling out names, I guess some people get missed. But again, if that's you and you only missed, one or two or a handful of classes, you're not missing out on much, and it's only extra credit as far as your grade is concerned. Had somebody just come in that Reigns, I'm sorry, no Reigns, Reed's online. Who came in? Case Bolt. Case Bolt, thanks. So Case Bolt is here, and it is 8:01, so everyone else is gonna be late. All right, I'm doing, speaking of attendance, slight word to the attendance change. So now, instead of words, I'm gonna say, all right, the next word for attendance is, and then I'm gonna ask you a question about something I just told you. So for example, if I was doing this previous in the semester, I'd be like, the definition of biology is the scientific study of life. And then I'll say something else, and then I'll be like, by the way, the next word for attendance is, give me the definition of biology. Does that make sense? I'm not gonna do like 10 minutes in between, that's asking too much. But I keep saying things, repeating myself, important things that people still screwing up. So I'm like, and I don't know how, like I'm saying it over and over and sending it in announcements and sending it in emails and posting it on Facebook, and people, it's like still people making the same mistake. So now I'm hoping this will help make sure that you understand some key points that I'm giving you. So yeah, be aware of that starting today. So pay attention when I give you a word or ask you for an attendance word. It's going to be more of an answer of a question. Excuse me, it's going to be a question of something I just gave you. And then, as usual, at the end, if you could give me one question from the material I just presented in the lecture, yeah, that's it. Even if you think you know everything I told you, you just write it to confirm it. Or pretend like you were in my shoes and you're writing a question that would be on a quiz. All right, lab. Check your pre-labs or your emails. You already know if you can come or you can't come. Can't come, I already posted it in the comments saying, hey, you didn't do the video or whatever the case may be. Or I sent you an email. I believe if you can't come, for almost everybody who can come, I'm pretty sure I left a comment that said, I'll see you in lab or something like that. This lab, unfortunately, is a longer one. There's a lot of waiting, so you have to set up a preparation. Then you wait for a lot of them. Not all of them, but some of them. So be prepared for downtime. So if you need to bring like earbuds or whatever, then I don't know, be prepared for that. So your pre-lab that's due Friday for pre-lab 4 is due on Friday as always. It's turned in via Google Docs as always. As usual, there's a little 12-minute video with questions at the beginning, and then there's a set of questions, and then just like this week, there's a set of hypotheses, right? And this time there's 4 sets of hypotheses, right? So you have video, questions, and then hypotheses, which hopefully makes sense by now because hopefully you're getting familiar with the scientific method. You have to do your hypotheses before you do your experiments, right? So the next word for attendance is telling me how many sets of hypotheses you need to answer in the pre-lab for pre-lab 4. That's your first word for attendance. Next, exam 1, I'll remind you because, anyway, it's due on Sunday. It's going to be done via my online, just like you would do a quiz, and also quiz 2 is due on Sunday. To remind you, those 2 quizzes, quiz 1 and quiz 2, are meant as your study guides for the exam. So you can take them as many times as you want. Your highest grade is the one that gets recorded. In theory, depending on which quiz you take, every time you take it, you should get a different set of questions, or at least have them in different order to help you prepare for the exam. And you'll have 50 minutes to take 50 multiple choice questions. If you need more time than that, let me know. Send me an email. I'll meet you online. If I can see you, if I can see your screen, if I can hear you. So if I know you're taking a closed book exam, I don't care how long it takes you to take it. Any questions about the exams or the pre-labs for that matter? I forgot to ask if there's any questions about the pre-lab. All right, independent work is now down to 14 points per page. It went down to 15 on Monday, it's now down to 14. Eventually it's going to hit what it says in the syllabus, which is 10. You've got one classmate who's almost done with independent work because she's been taking me up on these high points and been turning in some papers. So I recommend doing it. And remember, there's no length requirement for an independent work paper. You could just say, think of a question, answer it, right? I was wondering why dog poop smells worse than cat poop. I looked it up, and according to this source, this is the answer, right? Something nice and easy. So any questions about announcements or reminders? You guys have anything to announce? Any extracurricular stuff you should share with us? All right, then let's get started. And please, oh yeah, sorry, I know I keep saying this, but I didn't write it down this time, so I almost forgot to say it again. Please let me help you. Spend 5 minutes to meet me online. Say, hey, I'm about to turn in my pre-lab. Can you look it over really quickly? So I can just look it over. It'll be so quick. I'm like, oh, oof, man, I'm so glad you came because you missed this. And if you don't do this, you can't come to lab. So please, it's really quick and easy. Let me help you. Please let me help you. And of course, if you can't come to lab today, contact me and we'll figure out what you need to do. You still need to do the lab. We're going to collect the data on the board, so you can get the lab, the data from your classmates, and then you'll just have to do the lab on your own. Let me rephrase that. You have to do the lab not by coming into lab, because you're not allowed into lab, because if you're not prepared, you're holding your classmates back. But that being said, again, you don't have to do it alone. You can meet me online, and I can have a one-on-one lab with you, right? And say, all right, let's pretend like we're doing this lab. I'm gonna walk you through the steps. I'll tell you what happened, give you the data, tell you exactly what that data means. Then you can read the question, and I can explain really what that question is asking you. Let me do this for you. Let me help you. I know I'm getting some people seem like they're having a hard time. I'm really here to help. But anyway, if there's no questions about that, let's move forward. We need to get finished with this chapter, because this chapter is this is the last chapter for the first exam that is due Sunday. This is where we left off. We were talking about proteins. I mentioned previously that proteins are made up of a bunch of amino acids put together, right? So the amino acids are the monomers. The polymers are technically called polypeptides. We put a bunch of them together, we have a polypeptide. Once they're folded in the correct shape, we then consider it a protein, right? So here's just an example. This is an amino acid sequence of lysosine. Not that you need to know what the lysosine is yet. I'll teach you that later. So this is just the primary structure. This tells you, I know it's hard for you to read, but this just tells you each one. Like, all right, first it's this amino acid, then it's that one, then it's this one, right? That's all it tells you. But because of those amino acids, like we talked about on Monday, that causes it to fold into its certain shape. And of course, this is the same molecule, the same protein, it just shown in different ways. And again, more so with proteins than any other molecule, it's all about the shape. Like, it's all about the shape for almost all the molecules, but very much so with proteins. If it makes things easier for you, there's chargers, like you plug in your things in the back of the table, so it makes it easier for you. So any questions about that? As far as the exam is concerned, that's not really important. That's just a demonstration, a visualization of something I mentioned before. This will be important very later in the semester. Probably nothing on it on the first exam, but later in the semester when we start talking about mutations and how things work, this will be very important. If you have a slight change in the sequence of amino acids, that can have a huge effect, right? That can really affect the protein's ability to function. So again, like I said on that previous slide, you have this amino acid and then that amino acid, so on and so forth. And each one is different, so they act differently. Therefore, that causes a different shape for the protein. And if you just change one of them, you change the whole shape of the protein, and therefore you can change its whole job, its whole function. And this is just an example. And again, we're going to use this example later in the semester, but for now, you don't need to know about it. If you substitute one amino acid in the hemoglobin protein, you'll cause a whole disease. You'll cause sickle cell disease. And here's a picture of what I'm talking about. Right? You change this one amino acid, right? Everything else is exactly the same, but because of a mutation that you're going to learn about later in the semester, you change that one amino acid, and now the protein has a different shape. And because that protein has a different shape, that whole blood cell has a different shape. And because that whole blood cell has a different shape, now you have this whole disease, right? So again, the point here is the shape is very important. If you just change one amino acid, don't even get me started if you change more, but if you just change one amino acid, it could result in a huge change, which is basically what this slide says right here, but in writing. One difference is enough to cause a protein to fold into a different shape, which alters the function and, I should say can, not always, but can cause diseases, or can cause a disease. It's not always bad, but again, this is just a preview. We'll talk more about this later in the semester. Misfolded proteins are associated with a lot of diseases, including severe nervous system disorders and something called prions. So I'm going to give you some examples. I'm going to talk about prions here in a second, but that's another good independent work topic. Right? I just told you misfolded proteins are associated with many diseases, and I'm about to give you, well, I just gave you one example, which is sickle cell. I'm about to give you another, which is prions, but you can look up for other ones for independent work. I looked it up, and according to this source, here's some other diseases that are caused by misfolded proteins. All right, and I'm gonna put a big question mark next to this, because I'm pretty sure I'm not gonna ask you any questions about this on the exam. It's been a while since I looked at the exam, so I don't want to say that for sure, but let's talk about it very quickly. So if you look at a brain, you have all these proteins in the brain. Here, these are the normal ones. These little dark purple ones are the misfolded ones, and they're contagious in that they're misfolded in such a way that when they get in contact with the regular protein, then that regular protein is also misfolded. And that causes diseases like BSE, which is also known as mad cow disease, Kuru, which is the human version of mad cow disease, and that's spread from humans eating other humans' brains, which is another independent work topic, right? Very interesting. Fatal weight loss in deer, elk, things like that, which is common here in West Virginia, relatively common. But if I ask anything, like I said, I don't think I will, but I'm definitely not going to ask you to memorize the different types of prions. Here's something that's important, very important, and it'll be even more important later in the semester. A protein's shape is sensitive to its environment. And again, all molecules, it's all about the shape, especially with the proteins. The proteins are all about the shapes. So if you have a change in temperature, for example, all right, as you already know from discussions we've had previously, when I say the temperature is increasing, if you remember, what that means is that the movement of the molecules is increasing, right? So because of that, generally speaking, when you add heat to some molecules, basically, depending on how much heat you add, you're going to start breaking bonds. Because those bonds, yeah, they're held together, but you imagine physically, three-dimensionally, something just moving it, pulling it, and yanking it. Eventually, it's going to break those bonds, right? So that's how changing the temperature would have an effect. pH would matter too. I know no one's taught you this yet. You don't necessarily need to know it, but that red thing right there, that's an oxygen. That white thing there is a hydrogen. Here's something you do need to know for the exam. So this is a good little quiz. An oxygen has what partial charge? No, that's a good guess. So it's an it's a negative. And so it's a partially negative, remember? Because an oxygen has 8 protons, therefore the electrons love to be near it. Therefore, by process of elimination, we also know that hydrogens have a partial positive charge, right? But that's just partial. Now, here's another good thing that you need to know for the exam, because right now I'm trying to talk about how pH affects proteins. If something is acidic, it has a lot of which kind of ions? And I'll list them alphabetically. I'll give you your 2 choices. Those are your 2 choices alphabetically. There's H plus and OH minus. If something is very acidic, what does it have a lot of? And you need to know this for the exam that you're taking on Sunday, or that's due on Sunday. Well, if something is acidic, and remember you did that table, which is probably very boring and redundant, but that's kind of why I did it, because you had to type more, less, more than OH, H plus. So does anybody know if something is acidic, it has a lot of what? Hopefully you're just scared to be wrong.
That's me, I wanna say H plus.
Yay, H plus, good. So if you put this molecule, this particular protein, this is just one example, if you were to put it in something that was very acidic, those H plus molecules would, I don't want to say, they would attack, if you want to say it like that. They'd be attracted to that oxygen and vice versa, right? That oxygen would be attracted to that hydrogen, that H plus ion, more so than the hydrogen, because that's a full charge. That hydrogen's got a full charge. That H plus ion has a full charge. Likewise, if you put it in something that's too basic, this OH minus, right, is going to be very attracted to the hydrogen and vice versa. Because again, the OH minus, that's a full charge. That's a full 1 negative charge. That oxygen, it's only slightly negative. So again, if you change the pH too much, either whether it's too acidic or too basic, you're going to cause, in this example, the hydrogen bonds to unravel. Therefore, the whole thing's going to unravel. Therefore, you change the shape. Therefore, you change the structure. So any questions about that? Okay. Also, if I remember correctly, the only thing your book really talks about is the change in temperature and pH. But there are some other factors. Again, another good independent work topic. What are some other factors that can cause the proteins shape to change. Environmental factors. So again, protein's shape is sensitive to the environment, right? If you have changes in the environment, you change, it could have bad effects. So based off of what I just said, that's your next attendance word. What are one of the two things in the environment that I just talked about that could change, that would cause the protein's shape to change? And for those of you who came in late, you don't know this yet. Well, first of all, make sure you email me and tell me you came in late. Otherwise, I won't know. But the new attendance word now, instead of me just giving you a word, I'm going to give you some information. And a few seconds later, I'm going to ask you a question. So basically, you just give me the information I just gave you. Because just helping to prove that you're paying attention. Also, if you came in late, I have really important information about labs and exams. So either stay back and talk to me about it, or watch the video when I post it. But a lot of people are just making the same mistakes.
I'm so sorry.
Anyway, that's it for proteins. Anybody have any questions about proteins? Good. Next, we're going to talk about nucleic acids. This is the fourth and final category of biological molecules. I'm going to go through this one actually pretty quickly because you're going to have a whole entire chapter on this. So in my opinion, this is simply a preview. I went light on this. On the exam, there's fewer questions about nucleic acids as compared to the other four biological molecules because, again, this is just a start. You're going to learn so much more about this later in the semester. But anyway, nucleic acids, what do? You'll probably need to know this. They store information, and they provide instructions on how to build proteins. So that's the two main things they do. They store information, and they provide instructions for building proteins. So those are the two things you need to know. Now, some of them do other things, which is you get independent work done, especially RNA, for example. RNA does do those two things, but also some other ones have some other functions. You can look that up for independent work. In this course, you're only going to learn about 3 different types of RNA, the basics, but there's so many other different types of RNA, and they have a lot of different functions. So again, you can look that up if you want. Another thing you're definitely going to need to know, whether it be on this exam or later in the semester, the 2 different types of nucleic acids are DNA and RNA. So as a refresher to remind you, when we talked about carbohydrates, you needed to know monosaccharides, disaccharides, and polysaccharides, right? Those are the 3 different types of carbohydrates that you need to know. When we talked about lipids, we talked about triglycerides, phospholipids, waxes, and steroids, right? You need to know about all those, except probably not the waxes, but definitely the triglycerides and the steroids. Those are the 2 really important ones, and the phospholipids. Then we talk about proteins. Oh, I'm glad I thought about this because I want to reiterate this, or iterate it, because I never said it in the first place. So for the proteins, remember I did list them different types, like there's storage proteins and enzymes and all that. So for proteins, that's the one where you don't really need to know the different types. So of all the 4 different types of biological molecules, proteins, we're just sticking like proteins in general. Just know about proteins in general. I'm not going to ask you about the different types of proteins. It's the one of the 4 biological molecules where I won't ask you the different types. And then finally, here again, we're finally where we're at, nucleic acids. There's 2 types, DNA and RNA. Later, you'll need to know the full name, deoxyribonucleic acid and ribonucleic acid. But for now, the letters will suffice. DNA and RNA are the 2 types of nucleic acids. Any questions about that? All right. This one, as far as this exam is concerned, not very important. I'm almost positive I don't have any questions about this. Almost positive. Because, once again, you're going to have an entire other chapter about genes. So yeah, you're going to have a chapter where we really dig into DNA and RNA itself. Then we're going to have a completely separate chapter from that where we talk about how genes work. But what is a gene? A gene is a unit of inheritance encoded in a specific stretch of DNA that programs the amino acid sequence of the polypeptide. In other words, based on what you already know from today's discussion, a gene is something that's inherited. It just provides the instructions about how to build a protein. Because like we said earlier, just got finished talking about, right? You put this amino acid and then that amino acid, and the order in which you put those amino acids together dictates what protein it is, right? So it's the gene that provides those instructions. And it is something that you can inherit. So yeah, that's what a gene is. That's the book definition. So once again, for independent work, you can look it up. This is especially, I was telling you about, I didn't give any names, but you know who you are. One of your classmates is almost done with independent work. A large bit of it was doing this. Right? The book said this, my source said this, wrote a little bit about how they're the same and how they're different. Nice and easy, right? And it's good too, because that also helps me make sure you understand. If you write something wrong, I'm like, oh, well, I'm glad you made that mistake, because now I can teach you. But anyway, so these instructions on how to build the proteins on which, amino acid sequence to use, they are written in a chemical code that has to be translated. And we go from the nucleic acid language to the protein language. That one definitely, I'm not going to ask you about that. We're going to have a whole chapter on that for sure. So again, I might ask you about the definition of a gene or what a gene is. I'm definitely not going to ask you about this whole thing of going from nucleic acid language to protein language. That's going to be later. And as usual, when we're talking about definitions, unless I specifically say so, I don't want you necessarily to memorize it verbatim, right? So when you're studying, I want you to know what a gene is. I don't want you to just be able to regurgitate those words. Any questions about this slide before I show you a picture that's going to demonstrate this bullet point that will not be on the exam? All right, here's the picture. There's the gene, right? You got a long strand of DNA. We zoom into one part of it. And then that, from there, we do something called transcription that you can learn about later that you don't need to know. And we copy the code that's there onto this strand of RNA. Then we can take that RNA, the code that's in that RNA, and translate it into a protein. Bring me this amino acid, next bring me this one, next bring me this one. And remember, again, it's those amino acids that dictate what protein it is, right? Because there's 20 different amino acids. How you put them together in different combinations dictates what kind of gene you have. Anyway, any questions about that picture? Yeah. And remind you again, because I don't want to overwhelm you any more than you guys already are, this part will not be on this first exam. Got a whole chapter on this later. All right, so next. Yeah, you might need to know this for the exam. And again, I'll remind you it's a multiple choice, so there'll be no questions where I say, tell me all the things nucleic acids do. Instead, it'll probably be more like, I provide all this information and ask you, what kind of molecule is that? Like, oh yeah, that's a key macromolecule in the continuity of life. It carries genetic information, it carries instructions. So all those information, those are the hints I would give you. And then for the multiple choice, you would probably have something like nucleic acids or proteins or carbohydrates or lipids, right? So just to remind you again, hopefully to alleviate some stress, that's how the questions are going to go. Never am I going to ask you like, what is a nucleic acid, right? It's not going to be like that. 100 level class. Anyway, I think the only thing I really need to explain is maybe this first bullet point. What we're saying there by the continuity of life is, like I mentioned in the previous slide, nucleic acids are inherited, right? Genes are inherited. So that's how we have this continuity of life. So all that information from your mom and your dad, they combine to make you, and then you'll pass that information down to the next generation in theory, so on and so forth. And again, so this is a little bit, this is very redundant actually. Now that I'm thinking about it. There's no new information on this slide. It's just stuff I've said recently. It's written in a different way. Any questions about this slide? Excuse me. The next slide also just digs a little bit deeper into this. So again, this is not going to be on the exam, but it's an introduction and it's in your book. So I wanted to talk about it just in case you're following along in the book and said, why didn't you mention this? Well, I'm mentioning it and I'm going to tell you it's not going to be on the first exam. DNA, that is genetic material. It is found in all living organisms. And RNA, mostly used for protein synthesis. And DNA uses RNA to communicate with the cell. So once again, a big old X through here. Mentioned to signify the fact that I'm not going to ask you those questions on this first exam. And again, I have this little star here, right here. Like I said, mostly we use RNA to make proteins, but there are other functions, and you can look those up if you want. All right. So next, like I said when we started this conversation, for the most part on this exam, when I'm asking you the difference between the four major types of biological molecules, one of the things you need to know, when comparing and contrasting the four, is the monomer and the polymer, right? So when we talked about carbohydrates, the monomer was a monosaccharide, the polymer was a polysaccharide. Technically, lipids aren't monomers or polymers, so we'll skip that. Proteins, right? We said proteins, the monomer is the amino acid, and the polymer is the polypeptide, right? And we said that it was we'll leave it at that for now. Now, for nucleic acids, you need to know also, here we go. Nucleic acid, the polymer is called a polynucleotide, which is nice and easy. I wish they were all like that. Monomers are called nucleotides, right? That makes so much more sense. Just like the carbohydrates, right? The protein is a little bit more confusing, right? You have amino acids and polypeptides. It would be a lot easier if it was amino acids and polyamino acids, but it's not. So yes, you need to know this. So the next word for attendance is based on the last slide. There are two different types of nucleic acids. There's DNA and there's anyway. So very similar to what we talked about with proteins, right? Not only did you have to know the monomer and the polymer, but for the first time in our discussion about biological molecules, you also had to know the parts of the monomer, right? So when we talk about amino acids, right, we talked about that central carbon and the three different things that they're bonded to that are all the same. But then we said the most important part in our discussion anyway is that side chain that it's bonded to. Because that side chain dictates what amino acid it is, right? There's 20 different amino acids, and it's the side chain that defines it, right? Not the other three parts, because the other three parts are identical. So here, this is sort of similar. So the monomers for nucleic acids are, again, they are called nucleotides, and they have three parts. There's a 5-carbon sugar, there's a phosphate group, and there's a nitrogen-containing base. Notice I put sugar, phosphate, and base in bold. As far as I'm concerned, that's all you need to memorize. Because in the exam, I'm not going to try to get tricky and say, hey, what are the three parts of a nucleotide? And one of the options is a 5-carbon sugar, and the other one's a 6-carbon sugar, and I'm not going to do that. So if you just remember sugar, phosphate, and base, you're good. Now, it's the base that's the only part in the nucleotide that varies. In other words, it's the base that defines which type of nucleotide it is. Just like on the amino acid, it was that side group that dictated which one of the 20 amino acids it is. The base is the part that dictates and tells you what type of nucleotide it is. And I haven't introduced you to the other types of nucleotides yet, but we will. Are there any questions about this slide before I show you another picture, basically of this? I think this picture is from your textbook, and the other picture is from the old textbook, but it's all pretty much the same. So again, the three parts of a nucleotide: phosphate, sugar, base. That particular base happens to be cytosine. Not that you need to know that. It's just an example. All right, if there's no questions about that side, I'll show you the next one, which is another picture. Again, just an example, and it just looks different. Here we have the phosphate, the sugar, and the base. And in this particular example, the base is a thymine, or a T. So now finally I can tell you, like I said, for amino acids, there's 20 different varieties of them, right? For the nitrogenous base, if we're talking about DNA, this is all going to be on the next slide, so not to worry if this is a little bit sloppy. If we're talking about DNA, it can either be an A, a G, a C, or a T. And you don't need to know that yet, but you will later. But yes, there's 4 different types. So the same way for amino acids, you have 20 different varieties. Well, for nitrogenous bases, that too, but also for nucleotides, there's 4 different types for DNA. Any questions about this slide before I give you this particular information alone in writing. And again, just to remind you, those are just examples. You don't necessarily you don't need to memorize that that's thymine that we're looking at. You don't need to memorize that this is what a phosphate group looks like, and this is what a sugar looks like, and this is what a base looks like. You don't need to know that yet. You just need to know that the 3 parts of a nucleotide are phosphate group, sugar, or phosphate, sugar, and base. So again, if we're talking about DNA, it has 1 of 4 nitrogenous bases. And again, later you'll need to know that. If anything, maybe, probably not, but maybe, I'll ask you to know that A, G, T, and C are the 4 different types of nitrogenous bases for DNA. Almost definitely. I'm glad I saw that. That's a reminder. So this is from the old textbook. The old textbook went ahead and told you that, even though they get a little deeper later in the semester. Yeah, so those are the 4 different types, A, G, T, and C. And on the next slide, I'm going to show you pictures of what they look like. Not that you need to know what they look like. But again, if anything, definitely know that there's 4 different types of DNA nucleotides. Just like for proteins, amino acids, remember there's 20 different varieties. Here, at least if we're talking about DNA, there's 4 different varieties. Here they are again. A, G, C, and T. Later, you might need to know the names. Later, the fact that 2 of them have double rings and 2 of them have single rings, that'll be important later. For now, it's just a picture. A picture from the old textbook. So
You said the bottoms have what? You said 2 of them has double rings.
yeah, so 2 of them, and again, you don't need to know it now, but later this will be important. 2 of them have double rings, 2 of them have single rings. Later that will be important, but for right now, it's just a picture. So here's a good question for you. It's coming up on the next slide, and it is a question that you'll need to know for the exam. If we're bringing nucleotides together, or hit anything together, any monomers together, what type of reaction is it when we bring them together? Let me draw it. So if we have a nucleotide plus a nucleotide, and we get polynucleotide, you'll never see it written like this, but I'm just using abbreviations. A polynucleotide plus water, there's your hint. What kind of reaction is this?
Yeah.
Yay, dehydration. Yes, you need to know that. For this exam, anytime well, not this exam, for the entire semester, as far as we're concerned, when you bring 2 things together, it's called a dehydration reaction. So again, if you're bringing your monomers together, in this case, your nucleotides, you're making a polymer. In this case, polynucleotides. Once again, it's a covalent bond. You might need to know that part, so I'll underline it. What you don't need to know on this exam, but it will be important later, is that this covalent bond is between the sugar of 1 phosphate, my goodness, the sugar of 1 nucleotide and the phosphate of the other, right? But you definitely don't need to know that now. And again, just to throw this back, just in case you thought of this, because I just did. When we talk about amino acids, like we talk about proteins, like this thing is covalently bonded to this thing. I don't even know if I told you which part was bonded to which part, because it doesn't matter. You just need to know that the amino acids are the monomers. So I'm not going to ask you which part bonds to which part. But yes, these are covalent bonds, and that will be actually very important later. Because as you should know, and you need to know this for the exam, covalent bonds are the strongest of the 3 bonds that I've taught you. Covalent bonds are the strongest, hydrogen bonds are the weakest. That will be very important later in the semester, and that fact will probably be at least 1 question on the exam. So anyway, if we go from the sugar of 1 to the phosphate of the other, that gives us this thing right here, which is called a sugar-phosphate backbone. And again, not very important for this chapter. We're going to talk much more about it later in the semester. So again, if you have a sugar bonded to a phosphate, bonded to a sugar, bonded to a phosphate, it's these bases, again, the part that defines which nucleotides, so right, it's this nucleotide versus that. It's the bases that kind of hang off the backbone, right? So they're not really connected, at least not on these covalent bonds. They're just kind of hanging out there. And again, for now, not important. Later, that will be. Any questions about this slide? Nice and easy. So, the next word for attendance. You bring two things together in a chemical reaction in this course. What do we call it? What type of bond is it when we bring two things together? Or reaction, excuse me, what type of reaction? But don't say it, it's the attendance word. Yeah. Sorry, I wasn't clear about that. It's not your fault, that was mine. Anyway, and like I mentioned earlier, it's on the extra credit, so if you give it away, that's okay. Anyway, really quickly, just so you know, DNA is double-stranded, which means that those polynucleotides are coiled around each other to form a double helix. That will be very important later, not necessarily in this chapter. If anything, maybe you know the DNA is double-stranded. The rest of the stuff we'll talk about later. But probably not even that, at least not for this first exam. Again, like I mentioned earlier, the bases are on one strand. No, I haven't mentioned this yet. I'm sorry. I did mention the fact that the bases kind of hang off the strand like appendages. Well, when you have a strand, the base is hanging off like appendages over here, and then you have another strand with bases hanging off like appendages over here. It's those bases that kind of hydrogen bond to each other that keep those two strands together. And again, not important now, but it will be important later. Also not important now, but important later, is the A's always pair up with T's and vice versa. G's always pair up with C's and vice versa. Now this is just an introduction. So because again, I guess as far as the exam is concerned, this is a very unimportant slide, except maybe the fact that DNA is double-stranded. Any questions about that?
You said they always pair up, A's always pair up with
T's. Yep, G's always pair up with C's, at least when we're talking about DNA. What I haven't mentioned yet, but I might as well since you asked this question, RNAs are a little bit different. So everything I've been teaching you so far is like all about DNA. RNAs have some slight differences that would be very important later in the semester, not very important right now. The next slide I'm going to show you is basically a picture of what I'm talking about. It's going to show you the fact that the DNA is actually two strands of DNA. Like when you think of that picture of DNA, what you're actually looking at is two strands of DNA that have been hydrogen bonded together via their bases, and they also happen to be twisted. And really, as far as we're concerned, the twisting part's not even important, but it just happens to be, so we bring it up. But again, like I mentioned, important later in the semester is that T's and A's always go together and C's and G's always go together. But for now, not very important. Here you can see this sugar, phosphate, sugar, phosphate, sugar, phosphate backbone, right? Covalently bonded. And again, later in the semester, that will be a test question. Like what kind of bond holds a strand of DNA together? You don't need to know this yet, but later. Covalent bond. What type of bond holds two different strands of DNA together? Those little weak hydrogen bonds there. Very important later, not important now. So like I said, RNA is a little bit different than DNA. For the exam, you're probably not going to need to know this, but we're going to talk about it anyway. So really quickly, because there are nucleotides, they are just polymers of nucleotides, right? Whether we're talking about DNA or RNA, the monomer is the nucleotide, and the polymer is called a polynucleotide, regardless of which one we're talking about, DNA or RNA. All the bases consist of, still, the sugar, the phosphate, and the base. Whether you're talking about DNA or RNA, the parts that make up their nucleotides are the same. But there are some differences, and again, I'm going to go through them quickly because I'm not going to ask you on the exam yet. Later in the semester, this will be important. The three main differences are RNA has a different sugar, which is why I just focused on the word sugar. We didn't talk about the type of sugar, not for this 100 level non-biology major class. But just so you know, the RNA sugar is called ribose, not deoxyribose. Hence the name ribonucleic acid versus deoxyribonucleic acid. Also, there's a substitution in the bases. Yeah, so instead of having a T, RNA has a U. So RNA still has, there's still four categories, four varieties of nucleotides that it deals with. It's just one of them is different. Like I mentioned earlier, DNA is double-stranded. RNA, on the other hand, is single-stranded. Now, don't get me wrong, sometimes RNA, first let me just draw this stupid picture. I say stupid because I'm really bad at drawing. But there's a DNA that's not twisted, right? Those things are hydrogen bonded together, right? And we call that double-stranded because those are two different strands of DNA. Now, there are some instances where RNA kind of loops back on itself so it kind of has the same kind of look to it, but technically it's not double stranded. It's just a single strand that folded back on itself. And again, as far as the exam is concerned, not very important. Actually, not important at all. The next picture I'm going to show you is going to show you that other nucleotide. So again, the nucleotides are the same, other than the fact that RNA doesn't have a T, and in its place it has a U. So the next slide I'm going to show you is a picture of what a U looks like. Not that you need to know it, but once again, phosphate, sugar, base, right? A different sugar, not that that's important for you guys, and also it's a U instead of a T. Any questions about this slide? Sweet. All right, really quickly. That's it for attendance words. Other than, again, come up with one question from what I gave you. For those of you who came in last year, if you came in on time, you can leave. We're done. I just want to talk really quickly to the people who came in late because these mistakes are killing you. We already talked about it, so yeah, you can go if you've already been here. Attendance words change. We're going to talk about lab. You should already know if you can come in or not. If you don't, I'm going to stay here for office hours. Meet me online or talk to me. I'll let you know if you can come or not. There was a lot of downtime in this lab, so if you need to bring some snacks to eat outside or some earbuds or some cigarettes, whatever, yeah, for smoking outside, obviously, do what you need to do. Pre-lab 4. The pre-lab is due this Friday on Google Docs. It has a 12-minute video to begin with. Then you have a set of questions. Just like so far, this is just like this lab, right? Then you have another 4 set of hypotheses. 4 sets. And I know that seems like I'm being picky, but first you need to know what you're doing before you come in. And obviously, you know, because of the scientific method, you have to do your hypotheses before you do your experiments. And I want to apologize. I know some people are very upset about what's been going on, but you can't come to lab if you're not prepared. You're wasting your time, and because you're not prepared, you're going to be wasting your classmates' time. And also, one more reminder. I know you're busy, so I'm not saying this like I'm talking bad, but you're supposed to read the whole lab, right? Even if you're done with the pre-lab questions, you're supposed to read the whole lab so you know what you're getting into. So there's no way, for example, this week, you could have read the whole lab and not saw that there was other sets of hypothesis questions. Because it literally says, what are your hypotheses to this, right? Moving on, exam 1, due on Sunday via my online. It's 50 questions. It takes 50 minutes to take 50 questions. If you need more time, meet me online so we can do it together. And independent work is now down to 14 points per page. Whew. So, any questions about all this? And then again, like I said earlier, I think before the late people came in, let me help you. Before you turn in the assignment and just meet me online and say, hey, can you look this over really quickly? Because oddly enough, the biggest mistakes that are preventing you from coming to lab are the easiest ones to catch and say, oh, hey, don't forget the video. Or don't forget you have all 4 sets of hypotheses to do. So let me help you. I'm here to help. And really, when you think about it, it's only the pre-labs I'm getting picky about because you have to be prepared before you come to lab. The labs, whatever, turn them in late, that's it. So I'm done for the day. Well, done with the lecture. I guess I'm officially now starting office hours, and I'm just going to stay in here for office hours.
Have you graded the last labs?
Well, I think so, as far as I know. So what happens, and I'll double check for you. What happens is when you turn in the lab via my online, I get an email. It says, hey, so-and-so turned in the lab, and I click the link and it takes me to it.
Yeah.
I'm caught up with all those emails, but it doesn't always work. So I'll take another look.
Yeah, you graded mine, and I had to email it to you instead of submit it online.
Uh-huh.
And then you already had gave me a 0 for it, and then I sent it to you, or I submitted it online, so it might be kind of just confused.
Okay, that might be it. So if you've completed the last lab and you're ready for it to be graded, like, not the pre-lab, the entire lab,
Yeah.
just upload that to the to my online.
Yeah, I uploaded it after you said something. You said it had to be submitted, so I submitted right after you said that.
Okay. Yep, I'll double check.
Am I allowed to come to that?
Oh yeah, let me check.
Well, ask every time, 'cause I don't want to be like, no, you can't, that'd be embarrassing.
Yeah, it looks like yeah. I remember you missed some things, but then
Yes, those videos, I be trying to, I'll be trying to read too much and I'll be trying to catch those words, like, you'll say, if you read this, you will put a purple
Oh, right.
unicorn and stuff like that, like you said, video, like, what did he have on different
Oh,
today,
right.
and I was like, so, I
That's okay.
I'm,
You
because
gotta
I'm,
know.
your red, that's what it is, it shows up for me, I'm an old person. So red.
I understand.